Good afternoon all. So today in the methodology and instrumentation we have entered the new unit. We have completed the first unit, that is different types of equipments used in microbiology laboratory, and then we have entered into the second unit, which include microscopy. So first we can deal with the principles which are involved in microscopy. Why do we use microscopes? To see things, objects or organisms. I think one spelling mistake. Organisms that are not visible with the naked eye, and to study the morphological properties at micro and nano scale lengths. Then to collect images at high resolution. Then to observe live phenomena under microscope. That means live cells or chemical reactions which can be observed under microscope. So these are the main uses of microscopes. And this is the resolution of the eye, that is 0.1 millimeter. And this is the microscope which have 1000x, through which we can see the bacteria, human hair, majority of the cells. So our eye has poor resolution. In the case of electron microscopy, its magnification is 1000x into 1000x. So double of the normal microscopy, so that we can see viruses, macromolecules, atoms, etc. For example, DNA, organelles, etc. can be observed through electron microscopy. Next is the purpose. We already have told the uses. Now next is purpose. To become familiar with the history and diversity of microscope instruments. Then to understand the components, use and care of the compound bright field microscope. To study the components. To learn the correct use of microscope for the observation and the measurement of microorganisms. These are all purposes. So coming to introduction, the microbiology, the branch of science that has so vastly extended and expanded our knowledge of the living world, owes its existence to which scientist Anthony von Leeuwenhoek. In 1673, with the aid of a crude microscope. And this crude microscope consists of biconcave lens, which are enclosed in two metal plates. Leeuwenhoek introduced the world to the existence of microbial forms of life. Over the years, the microscopes have evolved from the simple single lens instruments of Leeuwenhoek with the magnification of 300. At that time there was magnification of 300. To the present day electron microscopes, which are capable of magnifications greater than 250,000. Next is microscopes are designated as either light microscopes or electron microscopes. The light microscopes use visible light or UV rays to illuminate specimens, and they include bright field, dark field, phase contrast and fluorescent instruments. You are familiar with this microscopy. In the last year you have studied in detail. Fluorescent microscopes use UV radiations whose wavelengths are shorter than those of the visible light and are not directly perceptible to the human eye. So shorter than visible light. Electron microscopes use electron beams instead of light rays and magnets instead of lenses to observe submicroscopic particles. Again coming use of microscope. That is to become familiar with the principles of bright field microscope, then familiar with the component parts of microscope, compound microscope, use and care of compound microscope, practical use of the compound microscope for the visualization of cellular morphology from stain slide preparations. So we are using, and the last year we have already taken a compound microscope from the lab for the visualization and for the study of the parts. Are you remembering? So next is the principle. The microbiology is a science that studies living organisms that are too small to be seen with the naked eye. Needless to say, such a study must involve the use of a good compound microscope. There are many types and variations, but all fundamentally consist of two-lens system. The two-lens system is a variable but controlled light source and a mechanical adjustable parts for determining focal length between the lenses and specimens. So this is the principle. The light rays passes through the ocular lens and they converge actual image through the objective lens, and then again they diverge and pass out through the slit. So this what happens, this eye, the rays coming from the eyes got diverged, and what happens, the light passes through the, so she has an imagination that, in this case the light, this is the rays coming from the eyes, and this may converge and the actual image is forming throughout the objective lens. This is the ocular lens or objective lens, and this moving through the second lens is objective lens. First one is ocular lens. So the light rays passes, converge behind the ocular lens, then they move towards objective lens. Finally the actual image is formed as a virtual image. That is the image which is formed throughout the ocular lens, and it is a virtual image, and that image is magnified by the ocular lens. Actual object is here. Okay. So another picture, that is transmitted light or bright field microscope. That is directly the most unusual way of looking at things. This microscope we have in our laboratory. Light passes through specimen, and from the specimen it is covered with a layer of glass. Specimen directly to the eye. So that is the principle. Now the lens have two focal planes. What is that? The rays, these are the rays that the rays entering the lens are parallel. These are the parallel rays. These are entering as a parallel, and these three rays converge on the opposite side of the lens as with the camera, and that point is called focal point. Rays entering the lens through focal point. So the continuation of this is here. The rays which are entering through the lens at the focal point, and they diverge and they emerge parallel on the opposite side of the lens as with the projector. Next is, what is the job of an ideal lens? To accept as many rays as possible from each point in an object, to reassemble all the rays from each point at corresponding points in the image, in such a way that the distance travelled by all the rays from each object about to its point, to its corresponding image point, it is the same. So they all arrive in phase. So the objective lens would like a projector lens and forms a primary image. And this is the structure. This is eyepiece and the image on the retina. This is primary image and this is specimen and this is the objective lens. So the ground glass on the top of the microscope tube. It is made up of ground glass. And the eyepiece acts as a magnifying lens. So objective lens works like a projector lens, forms primary image, that is 10 millimeter below the top of the viewing tube. And eyepiece acts as a magnifying glass and examines the center of this image. Next is, this is the components of a microscope. I think we have discussed in detail last year, so we can go through it. So this is what filter, filter mount, and this is what condenser. This is filter holder and this is that aperture, iris aperture, and this is condenser clamping screw. This is the lever which is pre-focusing, and here what happens, these are the eyepiece. Okay, then these are the three types of object lens, that is for 10x, 40x, 100x, but there are 4 objective lenses. One is small one, that is 4x. Then one is 10x, another one is 40x, and last one is what, 100x. 100x is oil immersion objective. So these are the objective lens and this is the eyepiece is here. Okay, I had mistakenly sent that here. This is the eyepiece and these are the objective lens. Okay, so these are the components of the microscope. I am not going in much detail. We have studied in the previous year. So we can describe some parts. Okay, first is about stage. It is a fixed platform with an opening in the center, and it allows the passage of light from the illuminating source below to the lens system and above the stage. This platform provides a surface for the placement of a slide with its specimen over the central opening. In addition to this fixed stage, most microscopes have a mechanical stage that can be moved vertically or horizontally by means of adjustment controls. Less sophisticated microscopes have clips on the fixed stage, and the slides must be positioned manually over the central opening. Next is illumination. The light source is positioned in the base of instrument. Some microscopes are equipped with a built-in light source to provide direct illumination. Others are provided with a mirror, one side flat and other side concave. An external light source such as lamp is placed in front of the mirror. For what? To direct the light upward into the lens system. The flat side of the mirror is used for artificial light and the concave side for sunlight. Next is Abbe condenser. This component is found directly under the stage and contain 2 sets of lens that collect and concentrate the light passing upward from the light source into the lens system. The condenser is equipped with an iris diaphragm. So it has an iris diaphragm. With the iris diaphragm, we move this lens, we can open and close. There is a shutter controlled by a lever that is used to regulate what amount of light entering the lens system. So we are using this iris